IB Bio, Cell Structure and Function, Part 6, focuses on the structure of the cell membrane. The essential idea is that the structure of biological membranes makes them fluid and dynamic. Here is the outline of all the movies available for studying IB cell biology. Use this slide to reference the part of the unit that you need to review. This movie is focused right here. It's the first of 2 movies on cell membranes. This movie covers basic membrane structure, while Part 7 looks at mechanisms by which materials are transported across membranes. Here is an electron micrograph with the membrane highlighted. The membrane serves to regulate what materials will pass into or out of a cell. The membrane is semi-permeable, allowing some substances to pass, while not others. Remember, the membrane has a 10 nanometer width. So even with an electron microscope, this membrane remains a difficult structure to see clearly. That scientists could not see the membrane well because of its small size resulted in various theories about its structure, some of which were inaccurate. More on that later. In this electron micrograph, you can see 2 membranes side by side. The scale bar shows 50 nanometers, and you can use the scale bar to see that the membrane width is approximately 10 nanometers. This is a diagram of the cell membrane, also known as a plasma membrane, and this image shows you basic membrane structure. What we have are phospholipids in a bilayer. The hydrophilic phosphate head and 2 hydrophobic fatty acid tails make up 1 phospholipid. In a bilayer, the hydrophilic heads are in contact with water both inside and outside of the cell, while the hydrophobic tails are in contact with each other to the interior of the membrane. The relationship of the phospholipids to each other and the phospholipids to water on both sides of the membrane provides membrane integrity. So here are the IB syllabus statements that kick off this movie. Describe the amphipathic structure of phospholipids and explain their arrangement in membranes. Explain how the hydrophobic and hydrophilic properties of phospholipids help to maintain the structure of membranes. Amphipathic refers to the 2 different affinities of the phospholipid. One end, the phosphate head, is polar, thus attracted to water, while the other end, the 2 non-polar fatty acid tails, are nonpolar, not attracted to water. Amphipathic. This diagram shows the fundamental structure of biological membranes. A phospholipid bilayer. Notice the polar phosphate heads are attracted to water on each side of the membrane, while the non-polar fatty acid tails interact with each other to the interior of the membrane, non-polar with non-polar. The relationship between water and the amphipathic phospholipids provides membrane integrity. So the phospholipid bilayer is the fundamental structure of membranes, but membranes have other components as well. Thus we have what's known as the fluid mosaic model. Here's the IB syllabus statement. Draw and label a diagram of the current fluid mosaic or Singer-Nicholson model of the cell membrane. Include the phospholipid bilayer, cholesterol, glycoproteins, and integral and peripheral proteins. Here is a diagram of the fluid mosaic membrane showing the phospholipid bilayer and proteins. Some of the proteins are integral, and some are a little bit more peripheral. For an integral protein, such as this one, this region of the protein would be nonpolar, and these regions, near the phosphate heads of the phospholipids, would be polar. Here is another diagram of the fluid mosaic membrane. In this diagram, we can see glycoproteins, carbohydrate chains attached to proteins. As well, we can see cholesterol embedded within the phospholipids. The carbohydrate chains lie outside of the cell, so this region of the image is extracellular. This region would be within the cell, intracellular. Here's a nice diagram that reviews all aspects of membrane structure that I've mentioned so far. We have the phospholipid bilayer with integral proteins, some of which have carbohydrate chains. Together, these protein carbohydrate complexes are known as glycoproteins. Some proteins are more peripheral at the edge of the membrane. We can also see cholesterol embedded in the phospholipids. Notice that this image suggests a role for membrane proteins, right here, as channels through which substances would be allowed to pass. More on this later. Here is a specific IB syllabus statement with respect to cholesterol. State that cholesterol, as a component of animal cell membranes, reduces membrane fluidity and permeability to some solutes. Cholesterol is a non-polar molecule composed of 4 organic rings. The liver synthesizes cholesterol, and we take it in as part of the food we eat. Cholesterol serves as the molecule from which our steroid hormones are synthesized, such as testosterone. This is a nice diagram that shows cholesterol as a non-polar 4-ring structure embedded into the phospholipid bilayer. Cholesterol serves to reduce the fluidity of membranes. As well, cholesterol reduces the permeability of the membrane to some solutes. As I mentioned earlier, the small size of the membrane at 10 nanometers in width inhibited our clear understanding of its structure. Early electron micrographs seem to suggest a membrane model other than the fluid mosaic model that I've presented so far. Here's the IB syllabus statement around this idea. Analyze electron micrographs that led to the confirmation of the Davson Danielli model proposed in the 1930s. Then analyze the falsification of the Davson Danielli model that led to the Singer-Nicholson model. This is the fluid mosaic model that I've presented so far. The Davson Danielli model of the membrane suggested that the basic structure was 2 layers of protein on either side of phospholipids, like a sandwich. Chemical analysis of membranes showed that they were composed of phospholipids and protein. So that evidence led them to this model. As well, membranes did act as barriers despite being thin, and proteins could serve as those barriers. Again, these 2 pieces of evidence were used to suggest the sandwich model of the Davson Danielli model of cell membranes. Protein on either side of a phospholipid bilayer. Electron microscopy became a new technique in the 1950s, and the early micrographs showed membranes as 2 dark lines separated by a lighter band. Proteins were known to appear darker than phospholipids. Thus, early electron micrographs seem to support the Davson Danielli model. Again, you can see 2 membranes here. Each membrane appears as 2 dark lines, thought to be protein, separated by a light band of phospholipid. These electron micrographs appeared to support the Davson Danielli model for the cell membrane. But a new technique, known as freeze fracture, provided evidence that falsified the Davson Danielli model. When membranes are frozen and then cracked open, electron micrographs showed the proteins to be embedded in the phospholipids. Here's a diagram of the freeze fracture technique with the micrographs seen below. You can see the membrane cracked open here, and the proteins are embedded in the phospholipid bilayer. You can see the proteins embedded in the phospholipid bilayer of the micrograph here. Now, the proteins are of varied size and shape, and this would not have been of the type found on the surface of the membrane as suggested by the Davson Danielli model. Here is the Singer-Nicholson fluid mosaic model of the membrane. You can see the freeze fracture idea right here. Globular proteins are present in the center of the phospholipid membrane. Right in here, the protein would be composed of non-polar amino acids. For these integral proteins, polar amino acids on the surface would stabilize the protein, while the non-polar amino acids in the center would stabilize the protein in the membrane. In other experiments, proteins marked with fluorescent dye were found to move easily within the membrane. These proteins were not static on the exterior of the cell, as suggested by the Davson Danielli model. The fluidity of the membrane, the movement of these proteins in the membrane, provided the name fluid mosaic model. Here's a scanning electron micrograph of the nucleus of a cell. We can see the freeze-fracture technique here, where the membrane has been cracked, and we can see the numerous proteins projecting from the interior of the membrane. Now, further evidence that falsified the Davson Danielli model was done with fluorescent dyes on proteins embedded in the membrane on different cells. So here we have a mouse cell and a human cell. We have membrane proteins. And these proteins were then exposed to fluorescent antibodies, different colors, in each of the two different cell types. And then the two cells were fused. And within a short period of time, we can see the mixing of the different colors of the fluorescent antibodies in the membrane. And this was suggestive of the fluid mosaic model, that the proteins embedded in the phospholipid bilayer were mobile and could move around. And again, this refuted the Davson Danielli model, which would have suggested that the proteins on the membrane were static and not moving. And the last piece of evidence that falsified the Davson Danielli model was the varied size and shape of the proteins that were seen in advanced electron microscopy freeze-fracture techniques. And this is not what the Davson Danielli model would have predicted. The varied size and shape of the proteins was evidence of the fluid mosaic model. Here is a comparison of the Davson Danielli model with the Singer-Nicholson fluid mosaic model. Study this image and be able to discuss the evidence for and against the Davson Danielli model of the membrane. Once again, we can see two membranes here. Notice the width is approximately 10 nanometers. The small size of the membrane made it difficult to elucidate its structure. It's also important to keep in mind that something as narrow as 10 nanometers serves as the barrier that separates the inside of the cell, life as we would define it, from the outside, from non-life. Here is an IB syllabus statement that I've touched on throughout this movie, but I will strengthen this in Part 7, the next movie. State that membrane proteins are diverse in terms of structure, position, and function. List the functions of membrane proteins. The functions of membrane proteins are truly the subject of the next movie. Strengthen your understanding of integral and peripheral membrane proteins with this slide. Notice the channel that passes through this integral protein. This image is suggesting a role, a function for membrane proteins. Here's a list of functions for membrane proteins. At this point, let's read through the list with the understanding that I'll be strengthening your understanding of these functions in the next movie and throughout the entire course. Here's the list. Membrane proteins can function as hormone binding sites, as immobilized enzymes, cell adhesion, cell-to-cell communication, channels for passive transport, pumps for active transport, and proteins for electron transport in the processes involved in mitochondria and the chloroplast. Once again, here are the roles for membrane proteins while you examine the Singer-Nicholson fluid mosaic model of the membrane. Here is a very nice diagram of a protein embedded in the phospholipid bilayer. You must remember that proteins are polypeptides that would wind through the membrane, just as this diagram suggests. If you look closely, you can see an opening that passes through the center of the protein, possibly serving as a channel regulating what comes into or might leave the cell. Membrane proteins can also serve as enzymes. This diagram clearly shows the relationship of the polypeptide to the membrane and shows a channel through the protein. That channel might be involved in what's called facilitated diffusion or active transport. More of these in the next movie. In this schematic diagram of a membrane with a protein embedded in the membrane, starting from time 1 and moving through time 4, we can see the membrane protein actively moving a substance across by undergoing a conformational shape change. ATP would be required. The molecule enters the protein, binds to a specific site within the protein. An energy molecule known as ATP changes conformational shape of the protein, and the substance is moved to the interior of the cell. One more time with a list of membrane protein functions. Hopefully, as we move to the next movie, you already have some strength with the roles of membrane proteins. Here's an image that might help you better see the roles of membrane proteins. We have active transport. Energy is required to move material into a cell. As an enzyme, where there's a series of reactions where the catalyst is the protein in the membrane. As receptors, this is a protein receptor for, say, hormones that have arrived to the cell surface. Cell-to-cell adhesion or cell-to-cell junction, connecting two cells together. Cell-to-cell recognition, where this cell recognizes this cell as the same or self. And then cytoskeletal support, we have proteins that attach to membrane proteins to provide cellular structure. In this table, we can see the various cell membranes listed along the left side. We can see the amount of protein as a percent of dry mass in this second column. Notice the quantity of protein to be found in the membranes of the cell. Membrane proteins are very important to the functioning of a cell. And that brings us to the end of IB Bio Cell Structure and Function, Part 6.